It's my honor to introduce Ruedi Aebersold as our third plenary speaker. So Professor Aebersold has a PhD in cellular biology from the University of Basel and is known to be a pioneer in the fields of proteomics and system biology. He's known for developing applications of proteomics in the field of biomedical research, like, for example, the isotope-coded affinity tag technique, which allows to identify new diagnosis markers for diseases and a series of computer programs for the analysis of proteomic data. So his research and teaching career has taken him to the US and to Canada as a postdoc in the California Institute of Technology, as an assistant professor at the University of British Columbia, and as an associate professor to the University of Washington in Seattle. After this, in 2000, he co-founded the Institute for Systems Biology in Seattle, and in 2004, he accepted a position as a full professor at the Institute of Biotechnology at the Swiss Federal Institute of Technology in Zurich. Since 2005, he now has been conducting research from the Institute of Molecular Systems Biology in Zurich. He's been awarded numerous prizes and distinctions, among which, for example, they are the Human Proteome Organization Award in 2005, the Buchner Medal in 2006, the 2008 award given by the Association of Biomolecular Resources Facilities, and the 2010 Otto Nägeli Prize. I could go on forever, but for example, last year in 2020, the Marcel Benoist Prize. He's also a member of the Leopoldina and the co-founder and scientific advisor of companies Proteomedics and Biognosis. So welcome, Professor Aebersold, and I'm looking forward to hear and see your talk.
Thanks very much for the introduction, and I'm happy to talk to you. I would assume that after this intense course, everyone is already a little bit tired. And I'm going to make it fairly easy, because I'm trying to tell you how you can get something for free. That's usually something people like. So let me start, and then you will see what I mean with getting something for free. I assumed when I was preparing for this presentation that since it's already Thursday, that you've been learning a lot about advanced computational techniques. Learned a lot about computational techniques to identify and quantify proteins and to extract information from these data sets. The generic format of many proteomic analysis is essentially a data matrix where a number of proteins, preferably a long list of proteins, is measured over a certain number of samples. And so I will refer to this in the following for N samples versus P proteins data matrix. I think this is an outcome of many proteomic studies, and of course you've learned how to identify these proteins, how to deal with missing values, how to quantify these proteins to basically get a high quality data matrix with which one then can work, for instance, in systems like Perseus. So since this is the course, I would like to start with a quiz. So I would like to ask, why do we analyze one or many proteomes? So here's a multiple choice. So we could say, because we can do it. Some might say it is fun to do. Maybe after going through the intricacies of all the tools, maybe you have increased or diminished fun. Or you might want to say, I really want to use this as in a competitive spirit to break the world record for most proteins identified or most samples processed. Some might need it for a degree, and some might say, we want to generate new functional insights, basically learn new biology from such data matrices. And I would assume that most, if one did such a survey, I would assume that most of the students, and in fact most of the scientists who analyze proteomes, would like to do this because or want to do this because they want to learn new biology. So let's look what we're facing when we want to study a biological system with proteomic techniques. So one of the units that we want to study, I mean, it can also be more complicated if we talk about whole organs, whole organisms. But one of the unit of life that we study a lot in life sciences is a cell. And we can view this cell under a microscope. And these are wonderful, wonderful entities, but they're also incredibly complex. So this is, for instance, a picture of a U2OS cells, a human osteosarcoma cancer cell, which has been continuously growing in culture somewhere since 1964. It is very small, and the amazing thing is that in this cell, in each of these living cells, thousands of concurrent biochemical reactions occur at any given time. They're all controlled, they're all concurrently active, and of course they change over time and or in the conditions the cell is in. They also determine the ensemble of these biochemical reactions, and some determines the appearance of and the function of the cell. We could also call this phenotype. So we know, of course, this is one of the central tenets of biochemistry, that these biochemical reactions are largely catalyzed and controlled by proteins, and I think that's the origin of why so many people are interested in proteins. So each one of these cells, this was measured a while back by Martin Beck and Alexander Schmidt and Johan Malmström, contains approximately 8 billion protein molecules, about the same number as there's people on Earth. So all these proteins are packed into this little cell. They have a particular function, and the ensemble of functions determines the appearance, the function, and the phenotype of the cell. So now when we do proteomics and use powerful mass spectrometric techniques, we would like to assume that we are, if we study proteins, we're close to the function. And now how can we determine then, how can we determine path from the molecules we identified to function? We could do this, what is typically referred as protein biochemistry, and it's actually very old science, and that we would isolate a particular protein. We would purify this to homogeneity, and then we start to do functional tests on this protein. So we, for instance, would determine that this protein is a protein kinase, that it is a protease, or has some other function that a biochemist in a test tube can identify, can measure, and can determine, for instance, catalytic constants. So this has been a very fruitful area of research, but we might immediately say, if we needed to do this on so many proteins that are in a cell and to do it on these proteins, it is basically an experimental dead end. And I think that's where proteomics comes in, that in the proteome allows us, the proteomic techniques allow us to measure a large number of proteins concurrently extracted from the cell, same types of cells in the same state of cells. So the objective is not to determine what each protein is doing by its own if it was extracted from the cell, but the objective really is to analyze the protein context, the context of the cellular proteome. So I think a typical question would be, what happens to the cell, to the rest of the proteins, all the other ones that are, that all these proteins are present in there, if in a cell, if a protein is missing, or is, for instance, damaged, or is overexpressed. So these are, of course, very typical experiments that a biologist might be doing. It might delete one or several proteins from the cell and then see what happens. And the proteomic techniques allows us to see the proteomic context, how the cell reacts to this perturbation. And I think this is a fundamental difference, which is not so often explicitly considered, that we are not just trying to measure as many proteins as possible, as an old biochemist would have done, and determine the function of these proteins, and then somehow piece this together. But we really try to observe the consequences of changes of the proteome in the context of the cell and in the context of all the other proteins in the cell. So the postulate would be that proteomics is a necessary science because even if we knew the biochemical function of all the 8 billion proteins by the biochemical method, we would not really understand the functioning of the cell because it is not just an aggregation of protein functions, it is the coordination of the functions in the cell, and that is, we expect that this is visible and apparent from the proteome. So proteomics is not just fast biochemistry, but proteomics really is the basis of a system science. So if we translate this this kind of definition into experimental data or experimental approaches, we would say we know, or we can say we have knowledge of the genetic proteome. So we can look this proteome up in numerous databases where now most proteins that exist probably in a human body have been enumerated. And you probably learned about these databases, and some have even an exquisite annotation of the function, the structure, and other properties of the specific proteins. So this is a great starting point because it gives us an outer boundary of the proteins that we can work with and then have to explain them in context. But where proteomics comes in is when we can compare the proteome not as a generic proteome, but in different, can determine it in different states, so that we can, for instance, say we follow the state of the proteome over time after the cell has been, for instance, hit with a pharmaceutical agent. Or we can follow the consequence on the proteome by knocking out or eliminating one of the thousands of different protein types from the cell, and we can see what happens. And the amazing thing is that if we do such experiments and if we follow the proteome, a lot of extremely useful information comes out. So I would like to illustrate this very, very briefly with a study that a postdoc, Martin Mehnert, has done in our group. So we take a cell, this is not U2OS cells, these were actually HeLa cells, and Martin selected, for reasons I don't want to get into, a specific protein. So this is a protein out of the 20,000 protein coding genes that roughly a human genome contains. He selected from databases, disease databases like Cosmic, a number of mutations that have been observed in the human population and that have been associated by genome-wide association studies with a number of diseases, in this case cancer. So he selected here 1, 2, 3, 4, 5, 6 different mutations, all which ones have been appeared in human beings, in the genome of human beings, and they have a link to cancer. He then engineered variants of this gene in which he mutated or introduced these particular mutations in a version of this gene and expressed this gene in a human cell among these other very, very large number of proteins. So he basically created a series of cells which were otherwise isogenic and identical, and they differed by only one amino acid residue in one gene that is expressed in this cell. And then he did a number of measurements. I don't want to get through the measurements, I just want to summarize the data. The measurements that he did, so I just summarized them, I don't go into how he did this, was the structure of the DYRK2 complex. So this DYRK2 kinase forms a complex with other proteins. And he measured by cross-linking whether or not the introduction of any one of these mutations changed the structure of the complex. He measured by APMS and BioID the protein interaction network around this DYRK2 complex. He measured the overall proteome by DIA measurement, and he measured the phosphoproteome. So this shows just to some extent representatively the results from these measurements. And it shows basically here that the different mutations, each row here would be one cell line which differs by the other by as little as 1 point mutation in the genome, would actually show a difference in the proteome, shows a difference in the phosphoproteome, and also show differences in the protein interactome. So what we learn from this is, first of all, that each mutation that was in that single gene in a different position of the gene left in the cell, in the overall context of the cell, a specific footprint, and this footprint is detectable at various levels of the proteome. So at the abundance of proteins, actually this was the level where the least amount of information could be extracted. At the phosphoproteome, because this protein actually tested is actually a protein kinase, and also in the structure and interactions. So we learn that we could make links. I don't have time to explain this, but some of these mutations affect particular biochemical functions which are very well known. So for instance, one mutation, but not others, affected the assembly and disassembly of the nuclear pore. And the third lesson we learned is that there's more to the proteome than sequence and abundance. And this is the topic, the lesson I would like to expand on in the following of this talk. So basically, we have now established that when we want to do proteomics, we're not just trying to measure as many proteins as possible, but we would like to do the measurements of the proteome in the context of a biological situation, for instance, disease and control. And we are measured because we have the capacity to do amazingly broad measurements. We can learn how groups of proteins behave. And so for the most part, to date, most proteomic data sets that are analyzed are these protein versus sample matrices, where we do not distinguish between different proteins that come from the same gene. We do not distinguish between proteoforms. We do not distinguish between modification forms, and we do not distinguish between the association state. So already these protein versus sample matrices are enormously informative, but we would even now assume or postulate that the further to the right we go along this axis of the central dogma, then the more complex the molecular landscape of a cell becomes. Because at the genome level, we have approximately 20,000 protein coding genes. We know that these, through various processes, for instance, alternative splicing, alternative translation, alternative modifications, can generate a vastly expanded number of proteins, proteoforms compared to the protein coding genes. And we also know, of course, that proteins associate to some extent combinatorially into protein complexes, and really we know that oftentimes the functional unit of the cell is a protein complex. So we know the further we to the right we go, the more complex the molecular landscape becomes. We also know that the further to the right we go, the closer we come to biological function, because we assume that these modules, the protein complexes, are very close to the phenotype because they are the units that carry out many of the catalytic and control functions of the cell. And it's also so that the further to the right we go, the less we know. We do quite well over here, we do quite well here, but we do, at least for some of the modifications like phosphorylation, ubiquitination, and so on. But there's, of course, many more modifications which are largely unexplored, and we do not really that well to determine interaction states, to differentiate the interaction states between cellular states. So my next few minutes I will spend on trying to show that out of the typical data matrix we generate, which is measuring a certain number of proteins in a certain number of cellular states or samples, that we can extract by using some algorithmic developments, we can make statements about changes or differences in the interactome or in the state of different complexes, and we can make statements about different proteoforms that are detectable, and in both cases we focus on differences detectable that are very likely to be functionally relevant. So we're not trying to establish as high a number of proteoforms that we can with analytical techniques. We try to focus on those proteoforms which we can identify and which have very likely a functional relevance in the cell. And this is, from an experimental biology point of view, interesting, because if you can be sure, or at least have a certain likelihood, that a different proteoform or differentially changed complex is detected, we can also then design an experiment to test really whether the function is affected. So the first attempt, or the first short story, is we would like to detect perturbed protein complexes from bottom-up proteomic datasets. So we focus now on the very right here, where we think we are very close to the biochemical function and phenotype, and then I will show with some results that this is actually the case, that such analyses are very functionally informative. So the research question we pursue is, can we infer changes in complex composition from number of samples versus number of protein data matrices, as we generally can produce and as you learned how to analyze in this course. So the study design would be, we select sample cohort of different sample groups, for instance, control versus disease. Could also be a time course, could also be different pharmacological treatments. We generate protein abundance profiles by bottom-up MS. We do this almost exclusively with DIA methods, but this doesn't really matter in this case. And then we compute changes in the modularity based on changes in co-expression covariance. So let me explain what I mean with that. So this is an example of such data where we measured the proteome protein extracts from NCI 60 cell lines. So the NCI 60 cell line is 60 cell lines that have been very well investigated. We extracted the cells and we subjected these to SWOS DIA mass spectrometry across the cell lines in duplicate. So what we obtained is basically 120 protein profiles, and now we extract from this data set, we extract the signals, basically the transitions that indicate the presence of two proteins in these cells, and these are subunits of a particular enzyme complex, and this is ATP synthetase subunit alpha. Doesn't really matter here, but it consists of two forms, the red form and the blue form, that form a heterodimer. So we can see that in most cell lines, the blue and the red curve follow each other quite nicely, and that the red is about twofold more intense than the blue. So we would assume that in these cells, these two proteins forming this heterodimer have a ratio of, let's say, 2:1 stoichiometric ratio. But we see also cell lines where this is not the case. For instance, this one here, but this one here has almost none of this protein. This one here, the ratio between the red and the blue is vastly perturbed. The same is this one here, where the blue is essentially missing. So we would conclude that in these 60 cell lines, this heterodimeric protein, which is a known biochemical function, it's a part of the ATP synthase, we would assume that in these cells, where the ratio is maintained, the cell is complex in one particular functional state, and in the others here, it is in a different functional state. Maybe it is inactive because one subunit is essentially missing. So now we can do this, of course, not just with one example. We can do this with all the proteins detected. And this is to show that and now we expand this analysis from these two proteins, which were chosen because we know some biochemistry about this protein, to all proteins that were detected. So we basically do to cross correlation of the abundances of the proteins detected, and the blue would be all proteins detected, and the correlation is centered around 0. There's basically no information in this correlation, and this is of course expected, because why would the abundance comparison from randomly proteins randomly selected have any kind of value? We can also use just the proteins from the proteins which are known to be part of complexes, proteins selected from the quorum database. And again, there's very little information in there if we randomly correlate them, because there's of course these proteins in the quorum database are parts of multiple different complexes, and correlating proteins between two complexes doesn't really yield any information. But when we take the proteins that are known from the quorum database, which are known to interact, and correlate the interacting pairs, we see that we now get a dramatic right shift of this correlation of protein abundances. That means that there is clearly information in the covariance of proteins that are known to interact. So Maria Bullian, she was a postdoc in our group. She established a computer program that basically implemented this concept. So she isolates from databases protein complexes, the proteins which you know can exist, and then she measures these proteins. It's a reference-based method. She would use a reference, for instance, a control, and a disease-associated samples, and she would establish basically a null distribution of this protein abundance covariances for the proteins in the null in the control case, and then ask whether there is protein covariances that differ from the null distribution, for instance, in the disease case. So if if this is the distribution where these proteins in normal tissue would covary, then if in a disease sample a protein ratio would appear out here, she can calculate the likelihood that this particular binary interaction is perturbed in the disease versus the control. So this then there is a lot of statistics behind that for multiple hypothesis testing, correction, and so on. But the effect basically is what Maria is doing with this algorithm. She does basically looks for cases where the abundance difference, the ratio of abundance for proteins known to interact, deviates from one group to the other, for instance, from normal cells to diseased cells. So we applied this then to a clinically actually challenging task. This is work from Tiran Guo and from Jing Zhong, who worked at the time, he worked in my lab and then went to start ProCan in Sydney. So this is a question of prostate cancer research, and like in many cancers, there is the tumor progresses through stages. So these have certain names or numbers, but basically go from low grade to high grade. And it is very clear if someone has a low grade prostate cancer, it is not particularly dangerous. If someone has a high grade prostate cancer, this is very dangerous. The problem is in this particular case, in a type of cancer, are the intermediate cases, because they're quite abundantly detected, and it is not known whether someone who has this intermediate score will progress to the more advanced life-threatening case, or will basically stay in this relatively non-dangerous intermediate state. So these are intermediate Gleason scores. They refer to 7A and 7B, and they can harbor aggressive or non-aggressive disease, and this cannot presently be distinguished, which leads to a lot of over-treatment on one hand, and of course to a lot of missed treatments on the other hand, which leads to death. So the question was, can we use this covariances analysis to distinguish aggressive from non-aggressive cancer? So we analyzed 290 patients in duplicates. From each patient, we analyzed tumor tissue and control tissue, and this created a fairly extensive data matrix of about 1,500 samples versus about 2,500 proteins, which were consistently detected by DIA mass spectrometry, actually through measurements at ProCan in Sydney. And then we applied this covariance analysis to this data set. So this is the entry point, is this matrix, and now we select from a database like quorum non-complex, and we asked, are in between groups in these cells, between groups that are represented in this data set, do we detect differences in the ratio of covariation between proteins in the complexes that are being tested? And so we observed in total 116 proteins which were part of complexes and that have highly significant protein pairs that have altered quantitative ratios between healthy and tumor tissue. So this is not so surprising. This is actually not so hard to detect, of course, or distinguish tumor cells from healthy tissue, but it shows that there is actually a signal that can be detected with this covariance analysis. Then it became more interesting when we started to look into specific subgroups. So this is, for instance, a protein which is called LDH, lactate dehydrogenase, and it consists of two subunits, an A and a B subunit. And so they form actually tetrameric proteins with two A forms and two B forms, and that's the catalytic unit that carries out the biochemical reaction of lactate dehydrogenase. So we see a progressive change, not in the abundance of these proteins, but in the ratio of covariance. So we see that low grades have one type of ratio, and then the ratio progressively changes as the disease progresses to high grade. So for instance, the 7A and 7B cases, which are the clinically critical intermediate Gleason scores, have a significantly and detectably different abundance ratio, whereas the abundance of the protein itself is not actually changing. So it's a reformation, a rebuilding of the protein complex. So now what is LDH doing? LDH is an enzyme called lactate dehydrogenase, and it is in a very central part of the energy metabolism of the cell. So everyone will probably recognize here glycolysis, and then glycolysis generates eventually pyruvate, and pyruvate can go two ways. It can go either be converted to lactate by the enzyme lactate dehydrogenase, or it can go into the TCA cycle. So depending on how much activity of LDH is present, either more or less of the pyruvate goes into the TCA cycle, goes into lactate. So it is a critical regulatory state, a regulatory enzyme for the energy metabolism of the cell. Now the interesting thing is that the LDH consists of these two forms, and one of these forms is a very fast enzyme, and one of the forms is a very slow enzyme. So depending on the ratio of the subunits in the tetrameric forms, it is either a blazingly fast enzyme, which basically mops up all the pyruvate, puts into lactate, or if it's slow, the pyruvate builds up and goes into the TCA cycle. So we detect by this covariance analysis, we can basically detect the catalytic fitness of this LDH in these particular tissue samples, and this is a control mechanism that is related to an effect that's very well known in cancer, metabolic effect, the Warburg effect. And so we basically come from covariance analysis, simply data-driven, to learn a biochemical finding that tells us that in this particular regulatory enzyme regulatory state, the metabolism is functioning different in the different forms of the different forms of the tumor. So to summarize this part, using algorithm to test protein abundance covariance, we can infer altered protein quantitative ratios in complexes. This is exactly the same data that you might have analyzed by clustering. We can simply do a different type of analysis on this data, and we observe biochemically relevant information. Altered complex composition is immediately functionally relevant because it means that the enzyme or the protein complex is functioning differently. If the composition is different, we showed we show now in this example a reference-based version where we have to tell it to look for difference between two groups, let's say control and disease. But we also developed the reference-free version of the algorithm, which is able to actually form groups. So you just give it an unbiased, just a data matrix, and it will form groups out of that look where the protein quantitative ratios look similar to each other, but different from other samples in the same cohort. And the reference-based version is more sensitive, but needs prior sample groups, for instance, case and control. The reference-free form is less sensitive, but requires no prior information of grouping. So basically, we get relevant biochemical information for free from bottom-up proteomic data sets. I'll go to the next. The second example, I have to check time. Okay, I think fine. And here we would now like to detect proteoforms and which have very likely to have a function in a biological system in the cell. So we're now in this area here, where we say that we have a specific gene that produces proteins through transcription translation, but each gene may produce different forms of the protein through processes like alternative splicing, protein processing, or to some extent protein modifications. So what are proteoforms? Proteoforms are variants that originate from the same gene, but they have a unique amino acid sequence or post-translation modification. So alternative sequences could be a protein that can either contain or miss a specific part, so here indicated in yellow, and this part might be, for instance, spliced out or spliced in depending on the condition. And of course, everyone is familiar with PTMs. Proteoforms can be detected, for instance, by top-down proteomics, but it is very difficult to know whether a proteoform detected is actually functionally relevant, and that's what we would like to address. Can proteoforms can they be detected that are functionally relevant? This is work from Isabel Bludau, who did this when she was still here in my lab. Now she is in Munich, and actually the work that I'm showing now just came out, I think this week or last week. So let me explain the issue, what she did. So she developed a algorithm called correlation-based proteoform assignment, or COPF. So let me explain the scenario. So let's say we have a number of samples, and we have measured multiple peptides per protein from these samples by, for instance, DIA mass spectrometry or TMT or whatever. If we had now 6 samples of one type and 6 samples of another type, maybe disease and control, we would see that these different peptides, each line represents one peptide of this protein, would have a different intensity depending on the ionization efficiency and so on. But they would basically follow a line here, so they would be similar between the samples of one group, but they might be different from the intensity in samples from the other group. And we would simply say there is a pre-mRNA. This forms an RNA, it's translated into a protein, this generates a number of peptides, and these peptides are measured, and depending on the abundance of this protein in the sample, the intensity is somewhat higher or lower, but consistent between the groups. Let's contrast this with a case where we have a protein that is shown here, consisting of blue and yellow exons, and now this alternative splicing leads to a protein that consists of only the blue exons or the blue and the yellow exons, and that are then detected. So if this protein is present in one sample, then we will see in the samples of one type, we see a certain number of peptides, the red and the blue, that are similar to the situation over here. But if we now have, for instance, in the other samples, a splice occur, that 2 forms are being present, only the blue ones are present, we will not see the yellow exon peptides, but we'll see only the blue ones. So this would tell us that we have a difference in these 2 proteins, the long and the short form, in these 2 samples. So this is basically the principle that Isabel is using for her data analysis. She detects peptides from each protein. She then does pairwise peptide correlation across all samples. Again, we have this peptide versus sample matrix. That is the entry point, and she does pairwise correlation peptide against peptide. And if she has in the green case here, basically the peptides would correlate with each other very roughly this similar way. Whereas if we have two forms of peptides from the two forms of proteoforms, we would see two clusters emerge, which then we know which peptides belong to the one cluster and which peptides belong to the other cluster. And then she developed the proteoform scoring function, which I don't want to get into. But basically, she developed an algorithm which can distinguish these proteoforms based on the co-occurrence or covariance of the peptides across a sample data matrix. Now, the sample data matrix we chose here was to separate native protein complexes by mild separation, by size exclusion chromatography, and then to measure the proteins in each size exclusion fraction. So we take a biological sample, we extract native complex, and we separate them. And from about 65 fractions, we then generate bottom-up mass spectrometric data sets. Again, we use DIA measurement. So the postulate that we have is if we detect two or more proteoforms of the same protein that associate with different complexes, we assume that the different proteoforms participate in different cellular functions. So it's very simply saying if we have two proteoforms along with the short one, the long one is here and the short one is here with a different complex, then we would assume that because they are in different complexes, they are doing different biochemical functions. So the data sets we generate are approximately 65 fractions of the size exclusion chromatography, and we detect typically from cell lines about 5,000 protein elution chromatograms. So this would be 5,000 curves here, where each one representing one particular protein along the axis of size exclusion, and the intensity gives us the intensity of this protein based on the peptides and transitions that are being detected. So this is a coherent, very informative data set that can be exploited for the detection of new complexes, for the detection of different complexes, but here we try to find proteoforms that associate with different complexes in these cells. So the experiment that we did, or that Isabel did and colleagues, was to take cells which are in different cellular states. So these were HeLa cells, in fact, which are either at interface or they are mitotic, so they're undergoing mitosis. We extracted native protein complexes from either one of these cellular states. We separate them by size exclusion fractions, and then detect the peptides which lead to the identification and distribution across the size exclusion fraction of about 5,000 proteins. And now to this data set, Isabel applied this Kopf algorithm. So she basically asks questions whether she can find a protein that might be a protein that can occur in, schematically speaking, in a full form with the yellow exon spliced in or the yellow exon spliced out. And we could imagine a scenario that where the long protein form is simply occurring as in monomeric form, it does not appear to be associated with a complex, and then the Kopf algorithm would identify that based on the blue and the yellow peptides and their elution profile in a single peak. We can furthermore expect that a protein, let's say the short form, a proteoform of the same gene, but missing one spliced out exon, might associate with a complex, whereas the long form would not. And we would see that because the Kopf algorithm would identify only the blue exons and find them associated with a protein complex eluting somewhere along the fractions. So this is basically the information that we tried to extract, and this was actually very successful. So from these roughly 5,000 proteins, about 300 were shown to have appear in 2 or more splice forms, in two or more proteoforms, and quite a few of those are assembly specific, some are assembly and cell cycle specific, or some are simply cell cycle specific, and some are neither. So what does this mean? If it's assembly specific, you would say we detect from a particular gene 2 or more proteoforms, and the 2 proteoforms are not assembling to the same complex. Assembly and cell cycle specific would mean we detect 2 or more proteoforms from the same gene, and these proteoforms are going into different complexes, and they go onto different complexes depending on the cell state tested. So this is, of course, very interesting, because any change in proteoform and its association under certain conditions to a complex is immediately suggesting a biological function that can be tested. So here is just one example. This is actually a case where we have, which was like the one schematically shown. We have a protein that can form two proteoforms by splicing. One form would consist only of the blue exons and therefore only get blue peptides, and one consists of the blue and the yellow peptides as the long form. And we detect very clearly 2 proteoforms here. This would be the interface. This is the mitotic state. These are 3 replicates of these particular samples, and we clearly detect 2 two complexes, one containing the red and the yellow and the blue, so this is the long form, and one complex containing only the blue form, only the short form of this protein. Both of these complexes are beyond the monomeric form of the protein indicated here with the red line, so we can say that both the short and the long form of this proteoform of this gene derived from a particular gene partition in a protein complex that is different from each other, and they don't really change across the mitotic state. So this would be a case where we have an assembly-specific distribution of proteoforms, and so that's the example I wanted to show out of these quite many really interesting samples we detect. We can also use this Kopf algorithm to detect proteoforms that differ by modification, for instance, phosphorylation. So, and we do this by detecting the absence of peptides, and we assume if a peptide is detected in one state but not in the other, the absence is explained by a change in this peptide, for instance, by modification. So we would have the size exclusion fractions here, and we would detect the pink and the yellow peptides in the one form. This would be naked peptides that are detected in this protein, and in the proteins phosphorylated, we would not detect the yellow forms because these are naked peptides that can get phosphorylated under certain conditions. So we would see a size exclusion fraction of the transition traces which follow only the naked peptides which are non-modified. So let's show an example. This is a cell cycle dependent phosphorylation of a particular protein. The biological function here is not really important. But we have here we have the interface state, and here we have the mitotic state. Each curve here is the elution of a particular peptide from this protein along the axis of the gene of the size exclusion. We see blue peptides, and they're designated as those peptides which we know that can be phosphorylated in the cell from sequence databases, for instance, phosphorylation databases. So we see that in the interface, the blue proteins co-elute with some of these yellow peptides, which are peptides which are from this ataxin protein, and we see that in the interface, the blue peptides are basically missing from this curve. So we would assume that in this in the two cell states, this ataxin protein is associated with a particular protein complex, and that it is associated in the interface in a form which is non-phosphorylated, and it is associated with this complex in that in the interface, in the mitotic state, the phosphorylated peptides are not detectable, so it will be likely be phosphorylated. So the phosphorylation state of this protein changes as a function of cellular state. So this can be extracted from this data set with this Kopf algorithm. I think there's a lot of really functionally relevant information to be detectable. So to summarize here, contrary to common belief, proteoforms can be identified by bottom-up proteomic data. Isabel developed this Kopf algorithm that identifies proteoforms from proteomic data matrices. We showed an example from sex-SWOS data, basically from native separation of complexes, because we wanted to detect proteoforms associating with different complexes. But the Kopf algorithm also works with conventional N or N versus P data matrices that are not that are built like case and control studies, for instance, by disease versus healthy tissue. So this I didn't have time to show that. So in conclusion, I would like to summarize that in this course you have learned how to analyze large bottom-up proteomic data sets, and you learned about the mass spectrometry, quantification, identification, false positive rates, missing values, and so on. And the main motivation, of course, to go through this really tedious work and complex work is to gain new biological insights. To date, proteomic data sets mostly consist of protein or analyzed with respect to protein identity and protein abundance information. And what I try to show here, that there's a lot of additional functionally relevant information that is present in these bottom-up data sets that can be extracted when suitable algorithms are developed and applied. And I showed two examples, one related to the detection of likely functionally relevant proteoforms, and one related to the detection of perturbations in the protein quantitative ratios in protein complexes. So the important thing, I think, the attractive feature of such type of analysis is that we don't need to do more work in the laboratory. The same data sets, as if they are well structured and of high quality, can be deployed to such algorithms, and so we basically get new information for free. I'd like to acknowledge Moritz, Heusel, Isabel, Bludau, Max Frank, and Robin Haven, who developed this sex-SWOS data protein complex analysis. I'd like to acknowledge Maria Boulian, Amir Banaei, and Chiara Owerks, who worked out a kind of hypothesis-driven and non-hypothesis-driven PQ protein quantitative ratio analysis algorithms, and the proteoform inference algorithm Kopf, which was mostly the work of Isabel Bludau with help from Hannes Röst's lab in Toronto, from Max Frank, Christian Dörig, Moritz Heusel, George Rosenberger, and Paola Picotti here in Zurich. That I would like to finish, and I'm happy to answer a few questions.
Thank you for this talk. I, as a bioinformatician, always think it's very nice if someone analyzes data very deeply and gets all the information that's there. So I think Jürgen had a question first.
Yes, indeed. Yeah, so first of all, thank you very much for the great talk. So this was really impressive. I just have a small question that is related to your first project of the covariance analysis of these complex members. Do you happen to have the corresponding transcriptomic data as well? And would one already see partially the covariance structure in the transcriptome already, or is it completely only emerging in the proteome?
So we have this information also, especially from these NCI 60 data sets, and also from others, and it is largely not visible. I mean, there is a tiny signal maybe, but it's really not something one could use. So this really is really apparent at the protein level.
Hello, thanks for a nice talk. I have a question in Kopf algorithm. So once we know that there is so there is a variance in the proteins, how do we know what caused this difference, whether it is a phosphorylation or if this is a proteolytic cleavage or is it a translational thing? How do you think we can identify that?
Okay, so first of all, I want to say that the COPF algorithm uses as a basis also for reducing false positives, it uses the co-occurrence of two events. So for instance, if there's a splice variant detected or a proteolytic form detected and a protein is losing a part through proteolysis, then the algorithm assumes that there is two or more peptides which show this behavior. Or if it's phosphorylation, it assumes that there are two phosphorylation sites, which actually quite frequently is the case, that phosphorylation sites tend to be clustered. So this is a caveat. It of course will miss some single phosphorylation sites, but from a point of view of statistical power, it was beneficial to force it to recognize two forms, two events, and then to combine them. So you ask, how do we know what the underlying difference is? So if it's naked peptides missing, for instance, through proteolysis, that the protein gets clipped in half or a piece is lost, or splicing, then we see that in that the specific peptide is missing and is detectable in one and not in the other. And then the algorithm has a segment where it maps back these peptides onto the amino acid sequence of the protein. So for instance, if you would find that there's three peptides missing in one proteoform and not in the other, and these peptides are all clustered very close to, let's say, the N-terminus, we would assume, although this is not proof, but we would assume that very likely the process that led to this pattern would be a proteolytic cleavage that the N-terminus clipped off. If these peptides were, however, like I showed in one example, be somewhere in the middle, then we would assume that it's most likely that this was originating from a splice site, alternative splicing. In the case of phosphorylation, then we have the option, of course, if we use DIA methods for data acquisition, we have to go back to the same data set and extract new sets of transitions. So I didn't have time to go into that, but in fact, in this phosphorylation example that I showed at the very end, Isabel went back and basically computed what the transitions would be for the corresponding phosphorylated peptide that is lost from the one sample in the form of the naked non-modified peptide. And if indeed she found then transition groups which correspond to the phosphorylated form of that peptide. So that's an advantage of DIA data. You can form a hypothesis and you can go back and look for that. So for instance, in the case of splice sites, you might be able to postulate where the splice junction was, and you could go back into the data set and extract peptide transitions that correspond to a peptide going over the splice junction. You might find it and you have a positive result. You might not find it, and then it simply would mean it's maybe not detectable or not present.
Thank you for the answer and the question as well. So for the sake of time, we will only have one more question, which will come from Jans Klinar.
Hello. Thank you, Ruedi, for a great and inspiring talk. I really like this peptide-centric approach. And clearly, you want to have as deep coverage and as deep coverage of the proteome as possible. So you fractionate 65 or how many fractions with your size exclusion chromatography. Why did you get only 5,000 proteins after all?
So the fractionation was not really intended to obtain deeper coverage. Of course, this is also desirable, but the intent really was not here to get deeper coverage. The intent was to get an additional dimension of the data, and that is the size exclusion dimension. So we know whether a specific protein is in monomeric form in this sample, whether it is inner complex, and we know the molecular weight of this complex, and oftentimes proteins are present in more than one protein complex. So this is the information we wanted to get. And so the reason we get that number of proteins is to some extent due to the instrumentation. Now there is more sensitive mass spectrometers. This was done on a QTOF mass spectrometer, a triple TOF. And the other is we eliminate all. The other reason is there's of course there's many more proteins in there, but we eliminate this protein if they don't show a elution behavior. So the protein is simply in monomeric form at the end of the chromatographic distribution, or bunched up at the very beginning when there's lots of proteins come out, which are very large complexes like ribosomes and so on. We don't record them there. We only show these 5,000 proteins are those where we have an informative elution profile for these proteins across the separation range of the size exclusion column. And the size exclusion column separation range can be chosen between different columns. There's various size ranges, but none will go from very small proteins to the very, very large proteins. So there's limits to the size separation range, and the proteins I mentioned are those that are in the useful range and not in the extremes of very small and very large, where they simply bunch up.